Okay, nomenclature. We're now moving into our acids and esters. I don't expect you to see any kind of nomenclature in the acyl chlorides, really even in the esters, you probably won't see those, or the acid anhydrides, other things like that. We'll talk about those in a little bit, but you probably won't have to worry about naming those. The two that you will need to know how to name are your acids and potentially esters. So if you have an acid, what you do is you change the ending, that E ending that would show up in an alkane or an alkene or an alkyne, and you change it to ic acid. Okay? Because acids are your highest priority functional group, those are going to be the ones at the end of your name. Those are going to determine the suffix. Okay? Any other functional group will get turned into a substituent, and it has those terms in parentheses coming after each of those other letters. Okay. Your esters change their names. They're very similar to acids as far as nomenclature goes. You change that E to an O8 to explain the carboxylate kind of functional group. That COO. Come on, pen. You're looking at this functional group, right, in both the acids and the esters. If it's an ester, then what's coming off of that oxygen? An R group, some carbon-containing structure. So what you'll do is name the structure as the O8, which includes all of this other piece. That'll get the O8 end, and then you'll start it off with a substituent on the O8, which would be whatever the R group was. Okay, yes, I'm going to look at an example here in just a second. Okay. Questions about what's up there? Just see some examples. Okay. I'm going to go ahead and name potentially two of the easier ones. So in our first structure, we go through, identify that we have a 2-carbon chain. What functional group do we have? We have a carboxylic acid. It's a hydrogen. So we have a 2-carbon chain. 2 carbons get referred to as ethane. So we get ethane, and then we have an acid. This is where it gets a little bit weird. An extra O sometimes shows up in there. Ethanoic acid. All right, so if you see that oic or ic acid, you're looking at a carboxylic acid. Relatively straightforward. Yes?
You count the carbonyl carbon?
You count the carbonyl carbon because it is part of the carbon chain, just like you did for an aldehyde and a ketone. Have any of you seen this structure before? Yes, every single one of you has seen this structure before, guaranteed. Do you remember the name of it? What was that, Twee? It's acetic acid. Why did I just call it ethanoic acid? IUPAC versus common names. Okay. There are several acids that have common names associated with them. Pretty much the only one that I know off the top of my head is acetic acid. The rest of them, you would probably be given that information, so you don't need to know what it is. Okay. As far as nomenclature goes, you would look at that and call that ethanoic acid, okay, based on our IUPAC rules. The bottom structure, are actually questions about that first one? Nothing. Bottom structure. Where's our longest carbon chain? It's the same 2 carbons. There's our longest carbon chain. Right, why did I not carry through to include those carbons? There's an oxygen in between, so I can't count that. All right? So if we went through and looked at this, what functional group do we have sitting in there? We've got our ester, so we could go through and refer to this as, whoops, I remember since we're at an ester, we won't call it an ic acid, but we'll call it ethanoic ethanoate. Is there a substituent on the ethanoate? What is that substituent? 2-carbon structure, we have ethyl ethanoate. Relatively straightforward, doesn't seem too bad. Have you guys seen this structure before? Yeah, I think you have. In fact, those of you in lab guaranteed have. Also known as any guesses? What's up?
Oh, does that say okay for this presentation?
Oh, the bottom right.
Bottom right.
The structure.
Oh, that says okay. Yeah, I'm going to make you guys name that one too. Ethyl. Yep. It's acetate. Where did the acetate come from? This whole piece looks like acetic acid. Okay, there's your acetate. Okay, so I'll give you some time to work on the other two on the far right. You should be able to name both of those. We're following the same general principles for all of these. Okay, and yes, I did make that top one a little bit more challenging. If you got questions, raise your. So for that first one, we've got a bute 4-carbon chain. We want to circle or highlight our main chain that contains our highest priority functional groups. So we will count the propyl to the left as a substituent on our main chain. So we've got that propyl there. Between our functional groups, we have a carboxylic acid, an alkene, and an aldehyde. Which takes highest priority? The acid. So that'll be our determiner on the end of the name. How do we name the alkene? We'd use butene, so we'd have buteenoic acid. But where's the alkene show up? At carbon number 2. Why carbon number 2? Never mind, it counts the same either way. So it's going to be at position number 2 based on the numbering from our primary functional group, which is at the carboxylic acid, carbon. So our alkene starts at position number 2. I know that's a bit weird because our numbering is going to start with the carbon of the carboxylic acid, but it is still part of the carbon chain. Okay. Yes, there is an added wrinkle to that, which we'll talk about in just a second. Where's our next functional group? Or where's the last substituent that we haven't mentioned yet? The aldehyde. How do we name an aldehyde? Al is if it's at the highest priority. It's not highest priority, so we can't use Al. It's oxo. Okay, where does the oxo show up? The terminal position, or carbon number 4. We can't get away with not numbering it and just say, oh, well, it's an aldehyde, we don't have to number it.
It goes in front, right?
We have to include that number because we can't distinguish an oxo from an aldehyde or a ketone until we have our structure drawn. So until we say it's at position 4, we don't know where that oxo shows up, and it could be a ketone or an aldehyde. Okay? Where does the propyl show up? Carbon number 2. As far as putting all the name together, which should come first, the oxo or the propyl? The oxo, because of alphabetic rules. So we'd end up with 4-oxo, 2-propyl, bute-2-enoic acid. Do we need to specify where the acid is in this case? No, because
It's the highest priority.
the highest priority, and there's only one of them. Okay? If there was multiple of them, then we'd have to worry about specifying.
You need to say that this is 6.
That was the added wrinkle. We do have an alkene. The issue with that alkene is that it does exhibit cis-trans isomerism. We would have to specify in our name which one we had. In this case, it is cis, which gives us Z. So we would say 2Z, 4-oxo, 2-propyl, bute-2-enoic acid.
Oh,
2 because we have to specify where the alkene was. Okay. Last one. I always thought it was weird to name these compounds until I actually learned the rules of naming esters. And I was like, oh, that's actually kind of straightforward. How many carbons do we have? We have 4, so we're looking at bute. Do we have any alkenes, ienes, or ines? So we just got the anes, so we'll go with butane. And then where do we go from there? Is it an acid? No, it's not an acid, so we absolutely cannot call it anic acid. It's an ester. Butan O8. What's the ester substituent? Potassium. So we end up with potassium butanoate. So we'll name the salts of our carboxylic acids just like we would name an ester. It's a little bit weird, but that's the way it works. Okay. Other questions? Just happened to remember that because I was saying butanoate and it didn't sound right to me. Butanoate, I'm pretty sure, is fair, but you may also see this one show up. Butyric, okay, if it was an acid, or we might see, I don't know if we'd see butyroate. But this butyr is the same idea, 4 carbons. Why they decided to use butyr, I don't know. But if that was an acid, it'd be butyric acid. Very rarely would you hear someone refer to it as butanoic acid. Okay. Why? Again, don't know. Accept it. Okay, I'm going to hit pause. Well, actually, no. We'll just talk through it up on the board. So a question came up with, what does the N mean? And presumably this is what you're referencing. If you see it floating in the front of a name.
Yeah, it was like N-phenol or like a benzene.
N-phenol?
Mm-hmm.
Yeah, I'd have to take a closer look at that.
Because there's a couple.
N typically does show up, and its reference is normal, okay, which sounds really weird. Why the heck do we use this normal term? Okay, it can show up in acids and bases as well. Well, you've seen T-butyl. What did T-butyl mean? It was tert-butyl, so we had a tert-butyl group. Okay, so it was a reference of how those 4 carbons were arranged. I don't think you'll see it as S-butyl or I-butyl, but that's sec-butyl and isobutyl. Again, we're taking those 4 carbons and we're arranging them in a particular pattern. Okay, that pattern is what we then memorize. Well, if N means normal, what's the most normal way that you can arrange 4 carbons? It's just a straight chain. Okay? That's what the normal is in reference to. Just a straight chain carbon structure. So saying N-phenol sounds a little confusing. I'd have to look at that after class.
Yeah, because
Okay.
there's like another one too, like N4 So there's a couple N's.
I'll have to take a look at it. Other questions? Okay. Done with nomenclature. Spectroscopy. When it comes to spectroscopy with our carboxylic acids, mostly this is a review, so hopefully none of this looks too familiar or out of the world to you. We have, within a carboxylic acid, we've got a carbonyl, so a carbon-oxygen double bond, and an OH. Okay, so minimally, within an IR of an acid, we would expect to see the carbon-oxygen double bond, the oxygen-hydrogen single bond, and the carbon-oxygen single bond. So 3 different signals. Why do we ignore the carbon-oxygen single bond? Shows up in the fingerprint region, so why bother to look at it if it's going to be in a dicey area to interpret? So if we go ahead and take a look at 3 example IR spectras. The first one I happened to look up, which I believe was butyric acid, you get a nice, pretty clean sample, okay, or spectrum. And you see what looks to be relatively neat an OH stretch. Okay? And then we also have a nice intense carbon-oxygen double bond stretch. Is there anything weird about that OH stretch? Might be difficult to see within this. Where does it show up?
Right at 3,000.
Where does the OH stretch typically show up?
33.
Up in this 33, 34 range. So why did it shift to such a low frequency? It has to do with resonance and conjugation with the pi bond. Okay? That bond, the OH bond, has become incredibly weak. Why is it incredibly weak? What's the name of the functional group? Carboxylic. What do acids do? Give up H+. That bond is incredibly weak because by definition it is an acid. That hydrogen does not want to be attached to the structure. So we've weakened the bond. Anytime you weaken a bond, what happens to its wave numbers in IR? It shifts to a lower value. Okay? So it makes sense to see that OH stretch show up significantly lower values. So if you suspect you have a carboxylic acid, we'd expect to see that carbon-oxygen double bond and an OH showing up fairly low, usually in that 3,000 range. The issue with that is that very rarely does the OH show up cleanly like it did in this example, okay, for your butyric acid. I tried to find some messier examples, and even these are relatively clean. Okay? We still see this really broad, nasty-looking kind of OH stretch that's centered around 3,000. It looks nothing like a CHSP3, nothing like CHSP2s, or like our aldehydes. Okay? So if you just see a mess in that range, it's probably due to an OH that's next to a carbonyl, meaning a carboxylic acid. Okay? So it's a combination of multiple pieces of information. You look at the next one, okay, it's even messier. Right? We get this real tall peak coming off the top of that, so we get this broad baseline. Okay? In general, what I would expect usually for an OH, if I drew over the top of this, would probably be something like this, where just that whole range, maybe some nice peaks coming off the top of that, that whole range just kind of almost looks like a flat line, but it's this weird lump that goes down on both ends. Okay, so it usually just looks like an awful, awful mess. Okay, if you see that awful mess, you probably have a carboxylic acid present. Okay, kind of makes sense? Yes? That difference is largely due to a process known as lability, or labile, meaning that hydrogen coming on and off the structure. If we can effectively freeze the hydrogen on the structure, we have that bond, we can see it. Okay? But if that hydrogen is coming off the structure, that bond is progressively becoming weaker and weaker and weaker, which means what happens to the signal? Progressively shifts lower and lower and lower, versus when it's directly attached, it's up at the 3,500 range. So what you're kind of seeing is almost, if you will, an average of all of the possible locations that hydrogen could be relative to the oxygen. Okay. So if we see a nice sharp peak for our OH, it's because the hydrogen is locked onto the structure. It cannot come off. If it can come off, that signal tends to broaden a lot, okay, and kind of disappear, which we'll see in the NMR as well. Okay. Not a big deal to know that, but that's kind of what's happening with it.
In a carboxylic acid, we would never see that. The pH is always going to be acidic, right? I mean,
It's,
there ever
well,
a point when we would
it's
drop an
always
H+?
acidic, but that doesn't mean it's in an environment where it can come off. If you're in an organic solution, okay, where there's no base, that hydrogen's trapped. Can't go anywhere. So typically when you're looking at these IR, when you see the worse or the unclean examples down at the bottom, those are typically because the sample has water in it. Okay? A very, very small amount of water, just enough that the hydrogen on the acid is now in an equilibrium coming on and off. Okay? Something basic is present that's allowing that shift. Which is why if you think about all the IRs that you've run where you've had carboxylic acids, they looked awful because we don't control water or account for the water presence in any of our samples in the lab. So we almost always have a little bit of water in there. When it comes to HNMR, we run into similar issues. In HNMR, really the only thing we're looking for is that hydrogen. It'll typically show up about 12 ppm. So it's the furthest downfield. I think it's downfield. Biggest numbers on our chemical shift. Nothing else shows up in that range. So you'll see something around 12, maybe up as high as 14. Okay? If it shows up at all. The issue again comes back to, is there something else in the environment that can pull the hydrogen off? Because if the hydrogen's not attached to that oxygen, can you see a signal? Nope, because it's not there anymore. Right, so only if that hydrogen is pretty much frozen to the structure where it does not come off will you see the signal for the IR. Right, but again, there's a lot of water present. Most cases, if you have a carboxylic acid and you run an HNMR on it, you're going to lose that hydrogen signal. You just won't see it. Okay. Your ACS test is standardized. They will probably show it to you if it's there, okay, or tell you it should be there. This again has to do with that lability. Okay. If the hydrogen can come on and off, that signal tends to disappear. And by disappear, it gets broader and broader and broader until eventually you just don't see it anymore. Okay. So if we had an alcohol, right, they said an alcohol went through a process where you shook it with D2O, okay, and the signal disappeared. Why did the signal disappear? Well, what is D2O? It's heavy water. So instead of there being hydrogens, there's deuteriums. Okay. For an alcohol, the hydrogen attached to the alcohol is a rather polar bond. It can exchange with the deuterium of the water, which means deuterium is now on the structure. Well, does deuterium show up in an IR? Or sorry, HNMR? Nope. So if you can exchange the hydrogen for deuterium, you lose the NMR signal altogether. As far as carbon NMR, would we expect to see anything exciting there? Think back to what the functional group of a carboxylic acid looks like. It does have a carbon, so we should expect to see something. It's going to be a carbon-oxygen double bond. We'd expect to see the carbonyl show up. Okay. But if we take a look at this spectrum, you notice that it's showing up about 180, almost 170. Whereas our standard range for our carbonyls, our aldehydes and ketones, puts them where? In the 200 range. So why is the carboxylic acid shifting so much lower? Because of the resonance. What is that resonance doing to the carbon-oxygen double bond? Weakening it. So our carbon starts to look more like a carbon-oxygen single bond than a carbon-oxygen double bond. And what happens? It shifts to lower frequencies. Okay? That is a popular spectroscopy question in the ACS exam, looking at how the frequency changes when you look at resonance or other contributing factors. Okay? Questions about that? Okay. So, what would we expect for carboxylic acids? How would they react? Like acids. Hence, we call them acids. The classic marker for a carboxylic acid, or any acid for that matter, is looking at the pKa. Anybody know the pKa of your carboxylic acids? Your pKa for your carboxylic acids in that 4 to 5 range, okay, which is relatively acidic as far as organic compounds go. I remember water in your alcohols are up at 16. Phenols are at about 10. Okay, so you're looking at probably the most acidic functional group within organic chemistry. Okay, so we've got our proton donors and our proton acceptors. Our proton donor, our classic example is again the carboxylic acid. The classic example of a proton acceptor, an amine. Okay, we're using a nitrogen with that lone pair. How do we know it's an acid-base reaction? What's that? Only hydrogen is exchanged. So if we look at the two reactions shown, which of those is an acid-base reaction? What happened in the top reaction? change the location of that hydrogen. Take a look at the product. What happened to the hydrogen? Added to the bottom. So didn't I exchange the location of a hydrogen?
You did, but you added something else. You did-
I did something else in addition to the transfer of the hydrogen.
Didn't you do
Okay.
addition?
I did an addition reaction in that bottom example. Okay. We don't look at Markovnikov or anti-Markovnikov in this case, just because there's no difference. For that addition, it is a Markovnikov addition. So that's good. Okay? So when we're looking at our carboxylic acids, the primary functionality that we talk about, and this is all chapter 19, is just that it acts as an acid. That's pretty much it. Okay? But if it acts as an acid, what else might we want to do? What else might we evaluate? That's a good test question. What's a good test question to ask about acids? Ranking. What's the most acidic? What's the least acidic? Okay, so when we're looking at the reactivity of our carboxylic acids, it's relatively straightforward. They act as acids. But now it's a question of which one acts as a stronger acid. Okay? And that ranking process is tricky, so you have to be careful when you go through and approach it. Okay? You should remember that these rules have been summarized multiple times before on how we can alter acid strength and base strength. What we end up doing is looking primarily at our conjugate base when we're looking at acid strength. If the conjugate base is more stable, then it is a stronger acid. Okay? How do we decide if it's more stable? Okay, we could look for charge first. Charge isn't even on this list. But if our structure is not charged after it's lost the hydrogen, does that make it more or less stable? More stable. Well, if it's even more stable, if there's no charge, that makes it a really strong acid. Right? Other things that we could look for. Look at the atom that holds the charge. If it is bigger, right, that's going to become a stronger acid. Why? Because a bigger atom can stabilize that electron density. Right? If we then look at electronegativity, the more electronegative atoms can stabilize the negative charge. Why? They're more electronegative. The whole point of electronegativity is the holding electron density. Right? After that, you can look at induction and you can look at resonance. Right? Both of those are a little bit trickier to deal with, but both of those are very common to show up on the ACS test. Okay? So you can fully expect to see a comparison of carboxylic acids between induction and resonance and try and decide which one is the stronger acid. All right, so we got 3 examples here. I'm going to go ahead and let you guys spend, let's give you 2 or 3 minutes to go through and attempt this. Why is the left more acidic? It's carboxylic acid. It's a good guess. I like that. Not really a guess, but it's good. Can you come up with a better explanation than it's called an acid?
Oxygen can stabilize the negative charge better than nitrogen.
Why can oxygen stabilize the negative charge better than nitrogen?
It's more electronegative.
It's more electronegative. Would we expect the one on the right to be acidic at all?
No.
That's a trickier question. Why not?
Because nitrogen.
Nitrogens are typically bases. Okay, so let's throw it in, make it even further. Which one of these is more acidic? Why? Try it a different way. Which one's more basic? Oh, there we go. Now we've lost a unanimous decision. Okay. What's the basic part for both of these structures? The lone pair on the nitrogen. There's resonance on that one, which then means it's more stable. Okay, so we aren't going to put on a hydrogen, so it's going to be less basic. Okay. If we looked at the acidity of these, okay, your amines are typically not acidic, even though they're very similar to carboxylic acids. Why would we not say, well, why isn't there resonance? Can I just pull off that hydrogen and do that? Yeah? But isn't that what I was doing with this lone pair anyway? Yeah. So both cases. We'd have a negative charge on the oxygen even with the carboxylic acid.
It's a formal charge.
So this is an interesting question. The reason why I asked it, because I always wondered, why is that nitrogen not acidic at all? Because to me, that is resonance. It has that option to do that, just like a carboxylic acid. But for it to do that, what is it actually disrupting? It's disrupting the other resonance structure. The lone pair with that nitrogen is already involved in resonance with that pi bond. If I pull off that hydrogen, to get it to do any resonance stabilization, I have to break the other resonance, which is not a good thing to do. So your amides are actually not acidic or basic. They're exceptionally stable. Right? Why is that a good thing? Where are amide functional groups found? In your body. Okay? It's what makes up amino acids. Okay? Proteins are made up with amide bonds. Okay? So your body has taken advantage of some very stable chemistry to facilitate and kind of keep that together. When we look at the next set, those three, which one's the most acidic? Which one? The one with 2 fluorines. Why? But so did the other one.
Does it better?
Why does it do it better? It does it more. Okay, so our most acidic one is on that far left. We've got our middle one, and we have our least acidic. Okay, so we've got strictly an inductive effect. Okay, nothing else. Very last one. I haven't actually worked this one all the way out. I'm not positive on it. Okay, what do you guys hear? I heard right. I think I heard left. So you said left. You said left?
Oh, no, I mean, right.
Okay, why do you say left?
It's less substituted, so it's not as like stable.
It's got the same substitution.
Well, I think it's like, it's closer.
There's the argument that I would try and pull with this. The nitro, being our electron withdrawing group, is closer. So if we look at an inductive effect, we would expect a stronger inductive effect with the one on the left. For those of you that said the right, what did you say? This is a questionable one. What does that mean? Why? What you'd have to do is look at how your electron withdrawing group draws electron density away. You could put a positive charge here, or a positive charge up there. Right? None of those look pretty bad, but if we look at the other one, our para-substituted issue, what happens there? Pull our electron density out, put a positive charge here, or a positive charge here. Which of those looks really bad? Do any of those look really bad? Positive charge near the carbonyl is bad. Why? That carbonyl is already really positive, which says what about the stability of that structure? Okay, it's not as stable, but what can I do with the electrons on this oxygen if I remove those? I can donate them in to start to stabilize it. Okay? So I'm starting to put a lot of positive charge near that oxygen. So that oxygen starts to share its electrons with the carbons instead of with the hydrogens, meaning it becomes more acidic. The electrons get drawn into the structure. So then the question becomes is what is the answer? It's a good question. I don't know. I didn't look it up. Okay. How would we find out? We'd look it up. You look up metabenzoic acid and parabenzoic acid and you just decide what's the difference. Okay? Would you see it on a test? No. That's why I'm not asking you on a test. I'm asking you now. Okay? So you need to start thinking about what things can contribute to your acidity. How are the electrons shifting around within the individual structure? How can we balance things out? Okay?
So which one?
Wait, that's not how you name it, is it? Because it has the nitrogen. Yes.
Benzoic acid is the primary name, or the, not IUPAC, but its common name. It's a common name. So meta-nitro, para-nitro, benzoic acid. And then as far as which one, you'd have to look it up. You're not expected to know that for the test. But what you're expected to start to do is to evaluate the things that contribute. Inductive effects, resonance effects. How do those things contribute? The rest of these slides are looking at synthesis of acids. So what I've gone ahead and done was gave you a whatever it is, a worksheet thing that has the synthesis of acids on it because I didn't make it on the slides and didn't make it in time, so I figured I'd print it out and give it to you. Also on that little handout is a bunch of multiple choice questions, right, to give you some practice with that. We are officially done, so I'm not going to go any further. But on Wednesday, we will start Wednesday off with answers to that little assignment. Okay? So if at any point you have questions about what happened within that little assignment, when you start Wednesday, you should come in and immediately write those questions up on the board so we can start to talk about them. Because if there aren't any questions, I'll assume you understand everything that's happening within them, and we will move on, which is exciting stuff.
So you still cover timing, right?
Yep.